Oh, sorry, I caught a little cold before coming here, so I will do my best to give this talk without any coffee. So I know, I think we all deserve a, okay. Yes.
Perfect.
Oh, is it? Oh, okay, got it. All right. Thank you all for having me. I'm really excited to be here with you all today. I think, as Memming talked or said in the introduction, we've been doing a lot of work on scaling up models within neuroscience, and so today I'll tell you about some of the different work that we're doing in this space. And so, thinking about how we can harness the power of scale of all of these different data sets that we have in neuroscience, bringing them together into one sort of common unified framework. And what we'll talk about today are ways in which we're seeing that these pre-trained models can then transfer to new animals across different tasks, as well as across different brain regions or even across species. And so kind of the motivation behind this work is, the brain, as we all know, as computational neuroscientists, is a very complex system. And when we're interested in building models that can go from brain activity to behavior, this mapping potentially between this complex high-dimensional system and the output or the behavior that we want to understand is, a very complex system. And so potentially, in order to actually learn that really complex mapping, we might need a sufficient amount of scale of data. So we need more data in order to actually form that mapping. And, we need models that potentially have enough capacity to be able to capture this complex relationship. And so, and we've also seen, of course, over the past few years with the advent of all these generative AI tools and ChatGPT, right? Like a lot of that came from just scaling up and having enough data to really start to learn the different relationships between, language or images as well. And so within neuroscience, we are interested in being able to test whether or not we can actually bring together many of these data sets into a common unified framework. And so, as we know, in neuroscience, we have really diverse data sets that are captured from different brain regions, while animals are doing different tasks or different behaviors, and often across different brain regions and circuits. And so, And so we're interested in, being able to take all these different data sets and to bring them together into a common and unified framework. And so kind of our first work in this direction was thinking about what are the architectures and what are the different ways that we need to represent this data in order to start to scale up. And so one of our first kind of attempts at this was Poyo, now, decent amount of time ago. And so there were a few different kind of components within this architecture that we identified that were necessary in order to support this kind of scalable multi-session training. And so the first step, in order to kind of prepare all these diverse data sets for training is to think about how do we actually go about tokenizing the data. And by tokens, I mean, within transformers in language, we'll take a sentence and break it up into words or even smaller portions of the words, and each of those serve as like a kind of piece of content that is fed into a sequence model like a transformer. And so similarly, we need a way to take the activity of many neurons that are spiking or firing rates of neurons, and to be able to also put them into this sort of sequence level structure that can then be processed by a transformer so that we can leverage these powerful architectures to scale. And so kind of there are two main kind of components within our approach for tokenization. In this case, we first started by considering actual spiking activity of neurons, but we can extend this also to regular time series like calcium activity, where we might have regularly spaced intervals of activity. But in this case, when we actually consider spikes, we came up with a tokenization that can kind of really exploit the temporal precision of the neural code, as well as the sparsity of neural responses. And so to do that, we introduced this tokenization where basically every spike within the population becomes a token that's fed into this sequence model. And so what I'm showing here is like an example where I have 3 different neurons or 3 units, each emitting spikes at some particular time. And now I can basically tokenize taking, let's see where we are, oh, right here. So these three spikes here, tokens, correspond to these 3 spikes, and then you have 4 spikes here. Okay, so now every spike becomes a token in the model, and so now the architecture is able to look across different spikes and across different neurons and try to understand how all this information can be put together into some sort of decoding of behavior, for instance. So the first thing is identifying this kind of most atomic unit of the neural representation, and that's the way that we want to kind of make tokens that are at that same level of information. And so in addition to the time of the spike, because we'll actually treat these as events in continuous time rather than binning the data. And then the other key piece is that we need to have some way of telling the model which neuron that spike came from. And so what we'll do is we're going to learn just a vector or an embedding for every single neuron that we see during the pre-training. And much like we could consider, like different words or tokens in language having embeddings that might be semantically similar. So like dog and cat might be mapped to similar parts within this token space. We also see this sort of unit embedding approach as being a way of organizing different neurons that might have similar function or maybe even have similar cell type properties into this latent space, where now neurons that act similarly might also have similar unit embeddings. So essentially this tokenized sequence is going to consist of the time and the unit embedding of each of the neurons that spike came from. And so what this allows us to do is now take a population of arbitrary size with an arbitrary number of spikes, and now turn this into a sequence, which is going to be the number of total spikes for the population. And these unit embeddings are going to be learned throughout training, and so the model has a way of kind of implicitly aligning all the neurons that it sees and their dynamics into a kind of common space. Okay, and so now that we've created, this potentially really long sequence of all the spikes from all these different neurons within the population, the next kind of ingredient that we think was an important part of the scaling work is actually, instead of now applying a normal self-attention operation, which would normally be done in just a standard transformer, which is going to look across all the different tokens, so all the spikes and all the neurons and their activity. What we instead do is actually compress this long sequence into a latent space, into a kind of lower dimensional set of learned latent tokens. And now what does that look like? What you can think about is this learned set of latent tokens will now provide queries to the high dimensional input sequence. And so this gives a pretty flexible mechanism for learning many different rules that the neurons might be kind of playing by. But so these queries could, ask questions like, how many spikes are there from neuron A, right? And it could only query a certain neuron over certain periods of time, or these latent tokens can actually attend kind of globally throughout the sequence, and they could look for overall aggregate patterns of activity. So this is a flexible mechanism that allows aggregation of different information across the input sequence. And more importantly, or another important aspect of this is this allows us to now have recordings that have different numbers of spikes, different numbers of neurons, and with different firing rates that can all be embedded into a common latent space of the same dimension. So now this gives us a way to work with sequences that can have really variable lengths, and then form this sort of compression into this set of learned latent tokens. So this is inspired by the perceiver I/O architecture. This is just the input, right, the encoder, where we're going to take this spike train sequence from all these different neurons from a given session, a given recording, and we're going to embed them into this latent space. And then once we're in the latent space, we can apply kind of normal self-attention and go through multiple rounds of processing within this latent token space in order to compute whatever functions of the input that our model is interested in. Oh, sorry, I don't have this final slide here, but after we go through those different rounds of self-attention, then basically we can apply a similar operation at the output. So at the input, the latents queried the input space for patterns of information they were looking for. And at the output, we're going to do the reverse, which is now we're going to query for some sort of behavioral response, or it could even be neural prediction, but we'll query at the output and ask the latent tokens to give us their answers. And so we have this kind of bidirectional both compression and now expansion into whatever output space of interest. And the first way that we applied this was actually in a behavioral decoding paradigm. So in that case, right, we'll take neural activity, we're going to compress it, and then our goal is to predict some aspects of an underlying behavior at the output. And in our first model, where we were able to apply this sort of principle of scaling, we started with recordings from primary motor cortex as well as premotor cortex across 9 non-human primates from 4 laboratories, so all using different sorts of manipulandum for recording, as well as different recording devices as well, some using spike sorting and others using threshold crossing. So the data set in terms of neural activity had a wide range of variability that just comes from the fact that everyone processes data differently and has different kind of behaviors or tasks or goals. And so in this case, there's 4 different tasks that underlie the pre-training set. So all of this was non-human primates doing different motor outputs and reaching behaviors. And you can see here there's diversity in the underlying complexity of the structure. So models, so a simple task could be something like a center out reaching task, where there would be 8 different targets that the animal would make reaches to and then come back to the center, as well as these much more kind of complex random targets where they have to move to any given position on the screen. And so we see varying levels of complexity in these underlying tasks. And so we built this model, we trained it on all of these different diverse data sets, and in general what we found is that the pre-training on more data actually does improve our ability to decode on any held out trial or held out individual. And so we really saw that this could be a way to achieve the sort of scale that we need to maybe solve really complex functions of brain to behavior. And so this is just a summarization of some of the results, specifically focusing on transfer across tasks. So here what I'm showing is just if I take a single session of this random target task, which is a more complex motor task, I fit a model on each of these sessions individually. So there's 21 of those sessions within this pre-training set. And then I just look at what is the average R squared in terms of my kinematics prediction. across all of those different sessions. And so I get, about a 0.84 R squared in that case. And now what we can do is say, okay, well now on those held out trials, what if we did the pre-training across all 21 of these random target sessions? So we see, a really significant boost in the R squared. And then finally, what happens if now we add in a lot of data from this simpler center outreaching task, which you might think like, oh, this is a more complex task. Would simple data sort of help it? But what we do find is that, when you're adding in more data, even if it is from a different underlying movement distribution, we can see that there's even more improvements in the decoding performance. And so this gives us some insights that, even across diverse tasks, across different animals, across different recording setups, we can, use these types of training strategies to be able to effectively do better at decoding from the brain than we would have thought if we only focused on that one data set at a time. And, now there's a lot of folks that are moving in this direction towards multi-session training, but at the time, the kind of industry standard is to just do a model per data set, right? Because at that point, we didn't necessarily have a way to combine all this into one large model. And so, yeah, it's really exciting to see that, some of these benefits can apply even across these diverse settings. Oh, my time. Oh, I don't have it. Okay. And then we can also actually, so this is all testing on some trials or sessions from animals that we had recorded from. So like we knew things about the neurons that were underlying these behaviors. But then we can also apply, okay, there. We can also apply this to a held out animal that we didn't see during the pre-training. And so in this case, what we'll do is we're going to have to fine-tune the model in order to figure out or identify where the neurons from a new animal kind of fit within this representational space that I described before. And so even on new animals that we didn't see during pre-training, we can apply this same approach. First of all, just even employing this POYO architecture on an existing neural latence benchmark data set in the community. We found that we, just with POYO alone, even on a single session, get a lot of benefits just from using this type of architecture. And then with our pre-trained model, we can then fine-tune it on this new data set and see even more benefits in terms of the decoding performance. So this shows us that in addition to getting kind of sliding off. In addition to getting scaling from within, we can also transfer across animals. And to really just see how far we can push this idea and whether or not transfer is even more general than, sorry, hold on a sec. This is coming off. Hair.
Mm-hmm. Yeah, can you hold your hair?
Here, I think I can do it actually. Okay. All right, this is a fun session. Okay. All right. Okay, so then we wanted to see whether or not these same types of transfer properties actually apply to even weirder situations. And so in this work, we that's joint with folks at Mila and Patrick Minot, and now has actually been extended into a recent work from Guillaume Lajoie and Matt Parrich's lab. So kind of expanding upon this result in a new paper at NeurIPS this year called Possum. So here what we did is we looked at, okay, so I have this pre-trained model across non-human primates doing different reaching tasks. And now can we use that as a pre-trained source and then fine-tune on very different data, in this case coming from a human that's actually paralyzed, so they're not able to make reaching movements, but still in the motor cortex. And in this case, they're asked to imagine writing different letters, and then the goal is to be able to decode the letters so that you could actually go from brain activity to text. And so in this case, this was the baseline that was provided for this data set. This is now looking at the classification error of that per letter classification task that I described, like which letter are you imagining writing? And now this is just looking at multi-session pre-training in the human data, so 10 sessions, so we can drive down the classification error quite significantly. And then here we're just looking at what is the benefit now if we take this pre-trained model from the monkeys and now use this for fine-tuning or as a starting point. And so you see, even if we're looking at different species, doing very different tasks, you still have this benefit of this pre-trained model. So there's a lot of interesting questions about like what has it learned, and maybe we can talk more about that during the panel discussion as well. What is really leading to this type of transfer and these benefits? And these are still open questions that we're excited to explore. So in a follow-up work, we wanted to also see whether or not these same types of strategies could be used in sensory processing, as well as in calcium imaging, which is different from the spike level tokenization that I just described. And in particular, when thinking about scaling, especially in calcium imaging data, some of the largest resources are often leveraging Cree lines, which are targeting specific populations of neurons or certain cell types. And so in this second project, we wanted to see if we could also build a model that could be trained across diverse Cree lines and even across different visual brain regions, and whether or not we can still see these same types of benefits in pre-training. And this was work that was presented at iClear this year, joint work with Crystal Pan and Mehdi Azabu. So to address this question and try to scale things up, also in this case in mice, we use the Allen Institute's visual coding data set. It's a passive viewing task, so animals were presented with any number of these different, both very simplified stimuli as well as more naturalistic scenes or natural movies, while recordings were being taken from any number of these 6 different visual brain areas, so primary as well as higher order visual areas, and across 13 different Cree lines, which would tag different subpopulations of neurons. And so one of the things that we needed to do to extend the model is to be able to take into account all these different data sets, is to also be able to decode many different pieces of information from the data set. So previously we had decoding, but it was all movement kinematics, and so it was kind of one set of queries that were being given to the decoder or to the encoder. And so now what we've done is we've just extended this to be able to do a multitask readout. And this kind of gives us a lot of flexibility to include data sets that all have neural activity. That's always the same common input, but they might have different tasks or different things that are being like either shown to the animal or different aspects of the physiology that are being recorded. And so essentially what we do is we just we extend that decoding framework that I talked about before, where we're going to query from the latent space, but now we'll also have a learnable task embedding that tells the model, okay, this is the thing that I want to try to predict. And so we can, in the case of the Allen Institute dataset, we can do this. We can do this in terms of what stimulus is it, so what is the orientation of this drifting grading, or what is the natural scene that you're being presented. So those are all kind of sequence level classification tasks. We say, okay, what is the stimulus that the animal is looking at? But then in addition to that, we also have the pupil location as well as the running speed. So these are dense time series of behavior that are also acquired at the same time as the stimulus presentation. And so now the model can like flexibly query, like, okay, now give me the pupil location, now give me the running speed, or what stimulus are you looking at? So all these tasks are then kind of all placed into the training framework and used during the training of the model. And so this is pretty cool because now we can combine different data sets that actually have different tasks as long as they have the same underlying neural activity. So the first thing is that we train the model to solve these 10 different tasks, including running speed, pupil location. And then across the board, we found that the multitask model could do as well as, or in many cases, surpass the performance of a model trained only on a single task. And so, that's perhaps, it's not obvious that a model could be trained on so many different things and doesn't lose some of its capabilities on any one task. So that on its own is kind of exciting, that we can have a model that can just like flexibly work on all these different output tasks. But moreover, we did find that like certain tasks like the running speed could be more significantly enhanced by the multitask training. And there could be two effects. One is, you have more data because you can now put in more recordings. But also, it makes a lot of sense that knowing things about the physiology could help you to understand the stimulus. So if the animal's in a very vigorous state, maybe that can give you more context to understand the stimulus. And at the same time, it could also go vice versa, where knowing things about what stimulus you're looking at might help you with understanding things about the pupil location, right? So all of these things can kind of work in concert. One thing that we found that we thought was really exciting as well is, so at the input of the encoder, we don't include any information about the Cree line or about the brain region that the recording is from. We just give the model the data. And we tested a few ways to actually incorporate some of that information and found that it actually hurt the performance. So what was really cool is we don't give the model any of that information, we do all the pre-training, and then we go into the latent representations that are learned by the model, and we ask, okay, can I actually understand what Cree line or what brain region this recording was taken from on the underlying latent variables? And so what we found is that, indeed, the Cree line can be effectively decoded from the latent variables of the model. And if we compare against a supervised baseline that's trained just to predict the Cree line, which achieves about a 56% accuracy, I think that this is on all of the classes. So this would be... Yeah, I forget if it was on the excitatory or inhibitory populations or on all. Sorry, I'll have to look at that. But yeah, so the baseline achieves a 56% accuracy. And then, interestingly, if we just look at the latents of our model only trained on drifting gradings, we actually do a pretty poor job of differentiating the different Cree lines. But then when we include all these different tasks, so it's able to see many different stimuli, naturalistic, as well as things like the running speed, et cetera, now we're able to, from those latent variables, get a very high accuracy of decoding those different Cree lines. And so this is cool because it also shows us that, and there's prior work that also shows that decoding different cell types can be enhanced by having more diverse recordings where you can see those neurons doing many different things. And so this sort of, this result also aligns with those findings where when you just looked at drifting gratings, maybe a lot of the Cree lines aren't actually visible or differentiable. But then when you add in more data across more tasks, we start to see these kind of properties emerge from the underlying latents. And we did some hierarchical clustering to also see if there was some organization in the latents that was sort of reflective of this structure, and found some nice kind of preliminary evidence that there was some organization within the latents that also had to do with their projection class information, so like IT versus CT or PT cells. Okay, great. Any questions? It gives me a second to take a sip of. Let me take this one. Oh, someone? No? Okay. Yeah. Yeah. Yeah, no, it's a great...
Could you repeat the question for
Yeah, so the question was just about whether or not these models could do some sort of in-context learning, or what would be the way that this might be instantiated within this, so you don't have to do the fine-tuning, et cetera. So the models that I've talked about thus far are not, they're not generative models in the same way that GPT is sort of trained towards this next token prediction task. So we haven't actually seen any in-context properties, but I think, yeah, it certainly is an interesting idea to consider. And I think once we have kind of these generative architectures that are implemented, one might be able to just put it in data and just use that as a prompt and already kind of see how the representation space is being impacted. Yeah, I think there's so many open questions here. So far, and we can have more time to talk at the end, I'll talk about this next piece as well, but yeah, so far, I think in language, next token prediction has been shown to be, a very effective pre-training task. But I think within neural data, there's still a lot of open questions about what is the right way to pre-train these models in a self-supervised way. But hopefully we can be moving towards these, kind of unfine-tuned situations where we can just prompt a model and already get some interesting inputs out. Great question, thank you. Okay, cool. So in the last part of the talk, I will talk about self-supervised learning, but it won't actually be yet achieving this kind of next token prediction. Okay, so what I will talk about is kind of extending on what some of the results that I just focused on, having to do with Cree line or cell type decoding. And so in this work, we wanted to build an architecture and inference system that would allow us to build representations of what neural activity from individual neurons looks like, and in a way where we could build representations of neuron identity that could then be read out towards different downstream tasks, like asking questions about what cell type or what Cre line is this particular neuron, what brain region is it situated in, and then also things about like this neuron and what makes it unique from the rest. So towards a cross-day neuron tracking. So as I just motivated, if we have ways to learn features of neuron identity, this could allow us to do things like classify different cell types, do brain region localization, or also do a cross-session neuron tracking. And this is work led by Vinam Arora in the lab. Okay, so just kind of putting this into pictures now, what we'd like to do is we'd like to take the activity of a population of neurons to be able to then, form neuron-level embeddings of all of the neurons within, and then from that, decode these different downstream targets like brain region or cell type. Oops. And some of the design principles that we used when building this model is, first, there has been a lot of work that's looked at cell type decoding, where you look at single neurons, right, and their activity patterns, and then understand, okay, what cell type is it? But what we wanted to do is to be able to build a system that can form a representation of that neuron, but also use the context of the surrounding population when making that decision. And we know that there's a lot of different microcircuits that have really tight coupling either between excitatory and inhibitory types. And so it seems likely that knowing what the other neurons are doing should also help you know something about what your role in the circuit is. So there's that. We want to use the population context. We want to be able to do it in a flexible way so that we don't have to assume that we have exactly the same neurons each time, or the same size of population. So you could have different data sets that might have different sets of neurons that it's looking across in the population. And then the second kind of design principle is that there should be some properties of that neural response and activity that are sort of stable even across different types of stimuli and different contexts. And so while it might not be consistent, right, like a neuron looking at two different stimuli could have very different response patterns, we'd like to try to find these kind of invariants that are stable across different contexts. And so to do that, we used a contrastive pre-training approach. And so what that does, or in contrastive learning, you'll take different views of some sample and you try to map them to similar points within the latent space. And so in this sense, or in this way, we'll basically take two different views, so chunks from the neural recording that are separated maybe by some random distance that's just kind of sampled throughout the training. And we'll take those two different views, and then for a neuron, for a given neuron, across those two different views, we're going to try to bring those representations closer, while all of the other neurons should be different, right? So really the only assumption is that if I see neural activity at two different time points from the same neuron, I'd like to move those representations close to one another. And we can do this, like I said, randomly by choosing, different jitters or different amounts of spacing between the different views. And actually, in the limit, and in some of the work that we've done, we do it over the whole recording. So it's not just the kind of very local, temporally situated context windows, but really it could be a very global property that you want to achieve. And this is achieved through like an info NCE loss. So we're using a similar objective to SimClear to bring the same neuron close while pushing the other ones farther away. Okay, and so we then, after designing this architecture and modeling framework, we tested this strategy in 3 different ways that have kind of increasing levels of complexity. So the first just being the case where we can pre-train on all of the neurons from different animals, but we do the pre-training in an unsupervised way using the contrastive learning first on all of the neurons. And then we have some labels on some subsets of those neurons as to what their brain region or what their underlying cell type is. And then we'll freeze the encoder. In all these cases, we're going to freeze the pre-trained encoder. And then in the first case, we'll do some training of a linear layer on top of the representations to decode whatever our objective is. And we have some neurons that are labeled that we can use to train that classifier. So everything's done in an unsupervised way pre-training, and then there's some amount of fine-tuning or some amount of labels that are fed in when you try to decode the thing of interest. So this one's the easiest because we've seen all the neurons in an unsupervised way, and then we just see some of the labels of the neurons in the second phase. So now this one is transductive zero shot. So we'll do the same thing where we've seen all the neurons in the pre-training, but now for some of the animals, we don't have any labels from them. So like some animals are labeled, and we'll use that to train our SSL decoder. And then there's some animals for which we haven't seen any labeled information. And then the hardest one is that we have done the pre-training on a set of animals and a set of cells, and now we have a completely new animal that we're going to feed into the model. So that's like that's the hardest case where we haven't seen any information from this new individual. Okay, and so this is just a summarization of the results and comparisons against some of the previous kind of competitive models for this. So we do this both on spikes, so here looking at neuropixels from the Allen visual coding data set across 3 different inhibitory types. And then we also have studied, in this case, spatial transcriptomics data sets, where you have different numbers of subclasses or very simple things like excitatory versus inhibitory cells. And so this is the result of our method. And so we can, in this case, test it in all these transductive or all the different cases that I just described. Whereas a lot of these methods don't actually apply in some of these settings, so you see some, missing numbers in those cases. And so, across the board, we, in some of these cases, we see kind of massive improvements in the decoding capabilities, and there could be a number of reasons. Like, these models are all trained in different ways, but many of them don't have a way of really using the population context to be able to form these representations. So in our previous model, LOLCAT, which is a supervised model for cell type classification, it's just looking at a neuron. It doesn't see any of the surrounding population, so that's like a kind of univariate approach. And in a more recent work called Newprint, they take some some features of the population response. So there are things like overall firing rate or other summary statistics, but again, it doesn't really have the richness of what the other neurons are doing as a way of informing those underlying representations. So through this kind of new architecture that we built for Nuclear, we were able to do this kind of population level inference in a more flexible way. So, really big improvements. I guess another thing here is in this visual coding data set, we're achieving like a 72% accuracy on the transductive case, but it's surprising even in this zero shot, putting in completely new animals, we actually don't get too much of a reduction in performance. So this is showing, at least in this setting, these models are pretty robust and can be applied to new animals out of the box without any fine tuning. And we see bigger gaps in some cases, and we think some of that could be also due to the size of these data sets. So these data sets are much smaller, and to kind of I'll get to the scaling next. But we also then applied this to brain region classification. All of these were neuropixels, where you'd span multiple different regions of interest. And this is just showing the latents that are learned by the model, where we do see this kind of nice clustering structure of different brain regions. And we can also test the model in the same way. So this is cool because we've done the pre-training, we have this model frozen, and now we can just decode many different aspects of what the neurons are about, or what they're doing, or where they're located, right? We're not retraining the model, it's just different downstream tasks that we can query. Okay. And so to understand a little bit more about the scaling, or how scaling is really impacting these models, we did this analysis here. So on the x-axis is the amount of labeled neurons, or the proportion of neurons that have been labeled out of our. So the results that I showed before would be this full amount of label ratio. And here I'm looking at the test performance across 6, 20, or 42 numbers of pre-training sessions. So this is just showing us that as we incorporate more sessions in the pre-training, and I'll also note that for this data set, when we have 42 pre-training sessions, that's also 42 animals, because the session is on a per animal basis. So we have, as we increase the number of animals in pre-training, we're seeing these pretty significant boosts in the underlying test accuracy on this cell type prediction task. And it's also showing us that you can have more unlabeled pre-training data that can benefit your cell type classification performance even more than getting more labels from some of those data sets. So this is really important because getting this labeled data is really costly and really hard, especially with electrophysiology, where they have to do optotagging experiments to get out the underlying different cell types. And so this kind of gives us some really important insights into how scaling and more unlabeled information can really start to unlock certain capabilities that we wouldn't be able to get even if we could really scale up the amount of labeled data. And we're seeing, maybe for this brain region prediction task, that we're kind of maybe saturating a bit, but you do see this gap, like if you only had 179 sessions versus 300. So I think this also shows us that we probably could go even further if we could have more unlabeled data, especially with this hard cell type prediction task. So all of these kind of works are, accumulating towards this greater goal of building foundation models for neural data. And so we really envision that, using all these advances in AI, the sorts of things that I've talked about, as well as, the field has made a lot of progress in this direction over the past 2 years. We would love to be able to really scale up even further, be able to add in more data sets of even more diverse natures. One could envision, a way of encoding neural data in such a way where you could actually flexibly accept many different modalities of neural recording. So most of what I've talked about today has been focused on electrophysiology or calcium imaging, but they haven't necessarily been put together. But we have some upcoming work where we're trying to do some of this multimodal neural recordings. But yeah, there's also a lot of information in local field potentials, or even if you were to, go even coarser, right, to looking at EEG as well as other sources of neural data. And all of these are giving us different pictures of what the brain is doing. And so potentially by accumulating information across all these modalities, we might actually get something that's greater than the sum of the parts. Adding in multiple species, because this gives us a way of doing comparative analyses of neural dynamics and activity. So, could we be able to see homologs or, certain information that's actually shared across different species through this type of pre-training approach? And we've done some work now on a bit of multitask, but still it's kind of focused on like within a motor system or within a visual system. And so it would be super cool to see how different brain regions across very different tasks could be combined, because all of this coming together gives us more insights into, what is the brain really doing. And then from this foundation model, we see that there's a number of downstream applications that could be enabled. I've already talked a bit about the BMI sort of applications, through this kind of brain to behavior decoding, cell type or brain region identification, but also, as a resource to the community, right? So if we have powerful pre-trained models that people can use as a starting point for their analysis on a new data set, this could be a really important way forward for the field. A lot of early, work in computer vision was enabled by just pre-training on all of ImageNet, and people were using that as a pre-trained model on very different medical imaging data sets, right? And so that's kind of the older generation of things, but I think even if we had something like that in neuroscience, where we had a strong pre-trained model that could be used as a starting point for different decoding or other analysis tasks, that could actually open up a lot of opportunities. How long or should I be wrapping up now? Okay. Okay, well, there's the conclusions then. That's good. Okay, so I talked about our POYO framework, the family of models, both for spikes and calcium in both motor and visual areas, doing multitask decoding as well. We talked about ways that we could transfer across tasks, some early evidence of transfer across species, across different brain regions. And then at the end, I talked about our new approach called Nuclear, which gives a way of reading out any of these different neuron level properties from self-supervised sort of pre-training. All right, and I would like to acknowledge the team and all of our collaborators. So Mehdi Azabu was really the lead of a lot of this early work on POYO, and then Vinam was also a big part of that, and was the lead behind the nuclear work that I talked about at the end. They also are the kind of, I meant to maybe put another slide on TorchFrame, but anyways, they also are like the co-designers of a new software package and kind of ecosystem that we've released called TorchBrain, which consists of a few different Python packages for scaling models, training different deep learning architectures on neural data. And one of the key things behind this is you can actually, we define ways of building like data samplers for model training that make it a lot easier for people to kind of plug right into this. And this allows you to actually pull down like little chunks of data from really long sequences without having to pull everything into memory. So this can also be a really exciting new way for people to work with long time series, multimodal time series. We're really excited to expand the capabilities of this and to grow the user base. And so if anyone's interested, please come up and talk with me. And also, there's more information here. And then, yeah, just like great collaborations. And yeah, and that's it. Thank you so much.